I have one extra topic. Am I good?
Oh, yeah, I think you're fine.
Okay. I have one extra topic I wanted to talk about that relates to experimental design before we move to the analysis. And this is because the extent of biological replication that is actually required for typical proteomic experiments is really underappreciated. And so we have seen already that if we have relatively small fold changes and relatively many proteins in the study, we need pretty large sample size. I don't remember what pilot study was here, but you see already that the number of replicates per group, if we have many proteins, is way above the typical replicates that people do. And even though I said earlier that I really appreciate the constraints of cost and so on, especially when it comes to predictive models, where you really want to do things like biomarker discovery, where you really want to predict the status of individual subjects, it turns out that for the moment, the extent of biological replication that people use is nowhere close to the type of replication that we actually need. And I just wanted to share with you one way for us to look into that. So this is now shifting to this objective number 3, right, where we try to predict the status of individual samples. So this was a particular case study. It was an SRM experiment where we had 200 subjects and 70 proteins. And these were, so this was subjects with colorectal cancer. And the goal was to use these 70 proteins as candidate biomarkers and see which of them are actually discriminative of healthy versus disease. And what was really unique of the study, and this is really how it should be done, is that the study also had another 260 subjects which were used as an independent validation set. So essentially, we use the first cohort to find this decision boundary, to find this classification rule, and then we use the second cohort where we apply this rule and see how well we do. And this allows us to evaluate whether this rule is actually working well in practice or not. And so the idea is, and this is a really large-scale study, so we could see how important or not the biological replicates are. And here's an idea. So if we really want to make, yes?
So in the previous study, when you have a training and validation sets, so what if in such cases we have no significance individually, but the combined we have significance? So how do we treat these?
Okay. So yeah, sorry, I kind of went a little bit fast on that. So here is the heat map. Remember in the beginning I was showing heat map, right, where the rows are proteins, the columns are samples, right, and the color is the abundance of the protein in the run. So this is this type of heat map. So significance, right, is associated with hypothesis testing. And hypothesis testing is a situation where we compare averages, right? So this was goal number 2. So now I'm shifting gears and I'm saying I'm actually interested in goal number 3, which is predicting the status of disease of individual subjects, right? Meaning that hypothesis testing and significance are not really relevant to this problem anymore. So what I'm doing, I'm starting from scratch, essentially, where I somehow summarize peptides and transitions of a protein in a single number. I haven't told you how, but let's say we have a summary. And so now this heat map has columns are protein, subjects, rows are proteins. Color is the abundance of that protein in the sample on some kind of summarized scale. So I'm actually not proceeding with hypothesis testing. There's no significance. I just say, okay, here's all the protein abundances, and I want to know out of those 70 proteins, can I have a small number, let's say 10 proteins, which combined can be truly diagnostic of colorectal cancer, kind of in the sense like Andy was talking about this morning. Can we have some kind of 10 protein rule or so, which will tell us who has the disease, who does not have the disease? So that's the idea. And for the moment, we don't know how many of those 70 are actually useful, right? So there may be much fewer of them, maybe none. But the idea, kind of focusing on experimental design specifically, we want to be in a situation where our conclusions do not depend on the choice of the individual subjects anymore. Meaning that if we add a few more subjects or remove a few subjects, it shouldn't affect our conclusions of which proteins are truly predictive. And this is exactly the process we decided to implement. So if we take this training cohort, and now if we take random list, include random list subset of these subjects into my kind of simulation. So we treat this cohort like as a population from which we can sample. We will fit some kind of boundary like that. So in this case, we used random forest, but you can really use pretty much anything you want. And we will see which of those proteins come as the top 10 most predictive proteins in this particular case. And we will report the accuracy, for example, number of true positives and true negatives among the total number of subjects, how they're classified. And then we will do it over and over again, and we'll do it 100 times. And then we will see what is the average accuracy, and we will also see here is these 100 iterations, and this is the frequency with which each protein was selected in this top 10 list. So ideally, we want to have some proteins with very high values and other proteins with very low values, right? So this is not something you would do necessarily in a clinical study, but it's for us to understand the importance of sample size. So we want to know how much the inclusion of individual subjects and the number of proteins affects our accuracy and affects the consistency with which the same proteins are consistently selected as predictive. So that's the idea. Okay, and so now we're doing that. First, we decided, what if we only have a study with a small sample size? Let's say we only had 10 subjects in the study. So we'll select 10 subjects from this big cohort, and we will select only 20 proteins, the most abundant, so with the highest intensity. It was an SRM study, by the way, I forgot to say. So the 20 proteins with the highest intensities in SRM, we calculate the average accuracy, it's 0.49, and the average number of times the best proteins are selected as most predictive. So roughly half. So we see that with 20 proteins, each protein at best is selected as predictive half of the time, depending on which 10 subjects are in the study. Now, what if we may increase the scope of our study and increase the number of proteins? We say 40 proteins and 10 subjects. Well, the accuracy improved slightly because we have more proteins, but you see now the number of times each protein is selected as predictive went down, meaning that now that the consistency with which each protein is selected went down. And now we went all the way to 70 proteins and see what happened. Our accuracy went down again. So we actually lost the accuracy, and we definitely lost the consistency with which we select the proteins if we increase the number of proteins only for 10 subjects. What it means is that we have too many proteins and too few subjects to make any reliable conclusions whatsoever. So now we can do the same thing and increase the number of subjects because we have many to choose from, and also increase the number of proteins up to 70. And so what you see is a very consistent pattern. So every time we increase the number of proteins somewhat, the accuracy improves, but then it goes down. Right here from 0.6 to 0.61, 0.5, right? 0.7, 0.75, 0.6. So usually when the number of proteins increases as compared to the number of subjects, the accuracy drops. And so the only way to kind of consistently have good predictions, so 0.8, and also consistently select the same proteins as predictive, is to increase the number of replicates by a lot. And this is another way. So what we did before was talking about hypothesis testing and false positives, but we can also use the same type of approach to understand how much the number of proteins plays out versus the number of replicates. And the idea is that we need a very large number of replicates if our studies have a large number of proteins. And we can do the same thing for different studies, but so now what we can also do, and this is what we currently implement in MSSTATS, we use this type of data as a pilot experiment, and we say, okay, what if we now simulate studies with even more biological replicates and potentially more proteins, what happens? And so we can make plots like that. So the x-axis is the number of proteins in the study. So 60 may be more common to targeted experiments, 5,000 may be more common to DIA experiments and so on, number of subjects. And we can simulate the data and see how consistently we select the truly predictive proteins in the study. And here the colors are the probability with which true predictors are selected. And you see that when the number of replicates is small and number of proteins is large, our chance of selecting true biomarkers is pretty much, it's 0.1, right? And the only principled way to do it consistently is to have a smaller number of proteins which we're interested in and a larger number of replicates. And we can do this over and over. We had many different data sets with which we did that. So the actual numbers change, but the conclusion is always the same, that the number of biological replicates we need is much larger than what is typically done in proteomics. And so this is just to attract attention to that. Okay, and so let me just kind of wrap up here. So the takeaways from the experimental design is that we need to define the problem, translate the biological goals into statistical goals, avoid vague terms. So often people say, yeah, I just want to find the signature of the disease. Well, I don't know what the signature means because it's not a specific term, right? So is it change on average? Is it prediction of individual samples, and so on? Experimental design, randomize replicate block, prioritize biological replication, and define analysis and protocol. So we will talk about this next. So do not cherry pick analysis steps, and understand the state limitations. So this is the part now that we will focus on for the second half. Okay, let me just, yeah, and this was the last thing. These are the points of significance vignettes. There are a lot more than here now, so this is a really good reading potential. All right, so now the second part, we will actually talk about how to analyze this data, right? So we spent a lot of time on the design, and I think it's really important. But before we do that, I actually would like to share with you another experience that we had, also our lab was part of, just to illustrate how challenging the analysis is as well. And unfortunately, this is a DDA example, so I don't have a DIA or SRM example, but I'm quite sure that the situation for SRM or DIA would be actually even worse than that. So this was a study by Association of Biomedical Resource Facilities in 2015. It's an association which compares essentially core facilities, so they compare good ways, good practice of conducting their experiments. And in this particular case, they focused on detecting differential abundance. And what they did was that they prepared a controlled mixture where we know the true change in abundance. They distributed the same mixture, analyzed it on the instrument. They distributed the data from this experiment to all the members, and they were all tasked to analyze that and report which proteins change in abundance among all pairs of conditions here. So here there was a yeast background, there was this protein spiked in different concentrations, and what we could give to the participants is several things. So either just raw data, and they would use their own pipelines, or we would identify SMS spectra, and they would just work on the quantification. Or we also used Skyline to actually identify and quantify peaks, and they would only focus on statistics. So they could choose from which part they start. And the question was then at the end, and it was all blinded, so we didn't know who is who. And then the question was to compare the results and see how consistent and reproducible specifically the statistics is. So everybody gets the same mass spectrometry files. And this is just to show the type of results that they had. So here the x-axis is the identifier of the study. The y-axis is the number of proteins that participants reported. Here they use the quantification based on peak intensity. Here they use spectral counting and some other things. Yellow means they started from raw data. Brown means they started from the peak intensities that we gave. And you see already here that number of reported proteins is not the same between participants, just in terms of ideas. These people misunderstood the instructions, so there's all kinds of things that can happen. So already there we see quite a bit of variability. But then the real interest for us was detection of differentially abundant proteins. And again, we have the x-axis is the identifier of the participants. The values below this horizontal line are the true positives reported by each study. There's at most 36 possible pairs of comparisons and proteins where these changes are statistically significant. So these were the numbers reported by different participants. The vertical line above the horizontal line, these are false positives. So they didn't know, right? We gave the database where the spiked proteins were obfuscated. And so you see, what you see is quite stunning, right? There's a group of people who are doing systematically well, and then there is roughly half of the submissions had a very large number of false positives. Here, someone had 2,000 false positives in this controlled mixture. This illustrates essentially how difficult it is currently for the community to come up with the consistent results of the same files. We're not even talking about different mass spectrometry, right? We're talking about analysis of the same data files. Now, here's interesting thing. So this is the absolute value of log fold change reported by each study, but only among the proteins which are the background proteins. So these are the proteins for which we do not expect any change. And so what you see that there's quite a bit of variability among background proteins pretty much for every participant. However, this group did not mistake some of this background variation for statistical significance, and they did well. So they could kind of distinguish random changes from systematic changes. This group was clearly misled by some of these false changes and thought that these were true changes. And this emphasizes how it's important it is to really have a procedure which is, consistent and appropriate for this type of data. And then what we try to do is to make some sense about what happened. How some people came up with many false positives and others did well. The participants had to submit a questionnaire which would essentially describe what they did. For example, the first column is the same as before if they used raw peaks or raw data or identified spectra and so on. And then we tried to break it down by how they did the identification, quantification, summarization, statistical inference, false discovery rate, and so on. Well, it is really to make any sense. There are different tools here. The point is that this figure has a lot of colors in it. So there are really many, many ways of going about this particular problem from the participant's point of view, right? And we tried, different fancy things. We tried different clustering and whatnot. Nothing really came out of that. So what we ended up doing is just partitioning the studies by positive predictive value. So this is the proportion of true changes among the reported changes. And so this would be roughly well-performing studies. This is medium performing, and this is or bad performing, and this is very bad performing, right? So less than 20% of reported changes are true changes. So according to positive predictive value. So that's what we could do. And then, of course, once we do this, all the people who looked at that, they were saying, okay, but which tool did better? Was it like, it's DDA, right? So did MaxQuant do better than Skyline, right? Or did something else do better? So can we kind of look into that? Well, so looking at the annotations, so here is all the groups which reported using MaxQuant and Perseus for DDA. Well, some did well, others did not so well. These are all the groups which use Skyline and some kind of modeling in R. Some did well, others didn't do as well. Some people decided to compare peak intensity versus spectral count. So these are groups which did both and both looked okay. This one found that one approach is much better than another approach. So even that type of conclusions depend very much on essentially what's going on. And so we try different things. Really, the tools there's no pattern related to the tools here whatsoever. The only thing we found was this. The first column here is the identifier of a lab. And the labs were allowed to submit as many results as they wanted. If they had multiple alternative ways of doing this, they could do. And here I circle the submissions from the same lab. And so what you see is that with one exception, these circles never cross this line. So essentially, the labs that have the expertise and who know what they're doing, they will use multiple tools and they will do well. And the labs that need training, they will use multiple tools and they still need training. So this was really the biggest conclusion. I guess it's not a surprise, right? But still, it's kind of interesting to see the empirical evidence for that. And this is why I think it's really important, well, to do courses like that, right? To kind of understand exactly what are the issues involved. And this is published, by the way, so if you wanted to use this data, you could use them now and try other methods as well.
I'm curious about the choice to rank based on the true positive as opposed to the false positive.
It's actually positive predictive value, so it's kind of both, right? So
Yeah, but I just, looking at that, as a biologist, I would rather have less true things and no false things. I'd rather...
That's exactly what it is. So you say, among things that you report as changing, what is the percentage of true changes?
Right, and I'd be happy with only 5 things being reported and missing 100 as long as those 5 were real.
I think because then the underlying data set is the same for everybody, I think these are comparable. But, you can kind of rank them based on this as well, right? And just see, okay, it was just a way of doing this. We were just trying to understand what to learn from that, just one way. But you can probably say, yeah, it's either the number or the percentage, you can kind of decide. We just felt it was the easiest way to put them together. Okay, So now let me tell you a little bit about what we do. And what my group does, we develop statistical methods specifically for this task. So we will not do things that Skyline is doing or some other tools are doing in the sense of, identifying peaks and locating the peaks, integrating the peaks, and so on, because these tools do it much better than what we do. We will take Skyline outputs, so outputs from any other tool. We'll use them as input to our methods, and we will focus primarily on statistics. So this is really where our contribution is. And it started quite a few years ago. It was a collaboration with the Ebersole lab, where the collaborator was interested in comparing in high-invasive and low-invasive breast cancer cell lines. But it was a kind of complex experiment because in addition to the cell lines, there was a different oxygen exposure, so normal oxygen versus low oxygen. And then this oxygen exposure was 6 versus 24 hours. And then also there were multiple these were cell lines, so multiple cell line cultures and multiple technical replicates. And what she was doing at that time is literally extracting individual runs and trying to do some kind of t-test for a subset of runs. She would be interested in questions such as, is the difference in oxygen exposure in this cell line the same as the difference in oxygen exposure in this line? And it was extremely challenging for her to put together all the peptides and all the ions together and kind of make some kind of sense at the protein level. And here is one protein from this study. Here the x-axis are all the conditions. Different colors are different peptides. This was a DDA study, and lines link a peptide. So you see that, for example, some conclusions are kind of trivial, so it's down-regulated in high-invasive cell line. But if you want to know what's the effect of oxygen exposure and how it varies by cell line, it becomes much less trivial because you see that peptides do not agree with them. These are zero values which become outliers. And so our task kind of came out of that, saying how can we work with experiments which can have an arbitrary complex designs and acquisitions which can have multiple layers of peptides or fragments or transitions, and also multiple layers of replication. How can we put together all this information and essentially make statements at the protein level? And that's really what our group set out to do. So since then, it grew into a pretty substantial effort for us. So at this point, what we do is relative quantification of proteins and peptides. So what's changing? We work pretty much with any arbitrary design of experiment. For a very long time, we were working with the type of data that with Skyline would support, so chromatography-based quantification, DDA, SRM, DIA, PRM, and so on. But in the last few months, we also have now MSSTATS TMT, so we also support TMT-based workflows. And we have multiple functionalities. So, of course, statistical modeling and inference, that's the core of this. But we also do data visualization and sample size calculation. And recently, we also extended that to things such as system suitability and quality control, so longitudinal monitoring, and assay characterization, finding limits of detection, limits of quantification. I may or may not have time to show this, but it's all published and out there, so you can take a look. And most importantly, we try to be inclusive. We try to work with as many tools that process the data in terms of finding peaks, identifying peaks, and so on. We have a really nice collaboration with the Skyline team, and MSSTATS is offered as an external tool in Skyline as well. But we also have converters to other tools, in particular MaxQuant, OpenMS, and commercial tools as well, SpectraNote and Proteome Discover, and others. And really, everything we do kind of starts from the tabulated data sets and tabular formats. And why R? And this is also another question which is asked a lot. So, well, part of this is just the bias of our training, because statisticians are often trained in R. But also from the user perspective, it's really lightweight, it's minimal, software requirements, easy to work on a laptop. For us, it makes a big difference because there's a large community of statisticians who develop various methods, and we can leverage libraries which exist for various purposes, which we do here. But also importantly, we feel like because this is a scripted language, so it's a command line language, it's really important for reproducible research. Because your script, your code, is essentially your lab book, right? Where you can document exactly every step that you do. And these days, R has a lot of infrastructure to mix text annotations with code, with the output. And it can really become a very thoroughly documented description which can be reproduced, which can be criticized, and so on. So we find that this is a very important component as well. Okay, so let me now tell you actually what we do. I spent like half of the afternoon here without sharing yet what we actually do here. So this is how statisticians view the whole process of conducting an experiment. And you see that this little cloud of the wet lab work is kind of in the middle. So what it means is that from our perspective, we have as much work to do before we collect the data as after we collect the data. It's really in the middle. So let me first talk about the data analysis, and then maybe I'll just highlight some of the things that we do here as well. So first of all, experimental design, and we already talked about this. We need to know what we're trying to do. Are we trying to look for differences on average? Are we trying to make individual predictions, and so on? What is the population? How do we define the population? What population is represented by our subjects? And then also make sure that we understand what were the strategies of randomization or blocking and so on, if we need to incorporate them. This was one example that I just talked about. This is another example just to illustrate the complexity of the design. So this was a collaboration with oncologists. They were studying osteosarcoma. This is a childhood cancer. They had 29 control subjects at a single time point, and then they had a time course profile of subjects with the disease. So they had plasma samples at diagnosis at various time points during chemotherapy, before surgery, after surgery, and then also eventually later. And the questions they would ask is which proteins change on average between controls and disease at diagnosis, which proteins are affected by chemotherapy, which come back to the levels comparable to controls after the treatment, and so on. So you can really ask fairly complicated questions from this complicated design. And that's what MSSTATS is trying to do. Okay, data processing. So we would work with the tools that do the work up to a point where we can export data into R in some sort of a tabular format. And this is an example of what the format looks like. Essentially, in this format, every row is a peak. In SRM, it would be a transition. In DIA, it will be a fragment. In DDA, it will be peptide ion. And it's annotated with a protein name, peptide name, charge, fragment, and then label. So for example, it's label-free experiment, everything is light. If it has labeled references, it will have light versus heavy. Condition, healthy disease or whatnot, right? Identifier of biological replicates, and then this is the intensity of the peak on the original scale. So either you have a tool and you can export data in this format yourself, or you work with one of the converters, or we can work with Skyline external tool to export the data in this report format. A really important requirement, which is important for us, is that each biological identifier, each biological replicate has a unique identifier. So that each subject is labeled in a unique way. And this is important because what we will actually do behind the scenes, we will try to infer the properties of the design. For example, if different identifiers are present in different conditions, we know it's a group comparison design, and it will require some type of analysis. If the identifier is present in multiple conditions, we know that it's a time course or repeated measurement, and then it requires a different analysis. And we will actually infer this behind the scenes from this assumption that each identifier is unique. Okay, and then the actual work starts, right? So data processing for us, the very first thing is quality control and normalization. And let's say this is one example. Here is a time course experiment with 10 time points. This is log of the intensity of the features, so peak intensities. And each box plot summarizes everything that was quantified in that particular run. So if you have many features, this will be all these features go in a single box plot. And MSSTATS does these plots by default. And what you see is that the intensity drifts upwards. Like here we have 3 replicates per time point. And the question is it a technological artifact or is it the real biology? And the fact is that it's really hard to know. unless you have certain things in place, right? And so that's really what happens. And for example, if we have some internal standards which were spiked in the samples, well, even better if you have some proteins which are not expected to change in the process, but those are very difficult to find. So if we have some internal standards such as IRT peptides, which are spiked in the constant concentration, or some other peptides used for normalization, what we can do, we can equalize the intensities from these standards across the runs and apply the same transformation to every endogenous peak in the run. And so if we start with something like that, we get, after normalization, to something which is more constant. And so this is the actual algorithm. We subtract the median of the log feature intensity of the standard and back the median of the medians just to bring everything on the same scale. So it is good because standards are not expected to have biological variation. It doesn't depend on how many features we have in the experiment. But it's bad because it only accounts for artifacts which happen after the standard was spiked. If there's some difference in how the samples were stored before or handled, we will not be able to correct for that. And standards can be noisy. So if we have peak overlaps or some other issues, if we try to normalize with respect to something noisy, we can do worse than better. And so usually what we came up with in terms of best practice is to use multiple standards, but not use all of them for normalization, but set aside some other standards and just treat them as if they were endogenous intensities. And then after normalization, you look at those standards specifically. So if their profiles got corrected, it means that the normalization worked. And if it goes like that, it means that it was a problem. So that's really what we use for those. And then another approach, if you don't have standards, another approach is to equalize the median of those boxes between the runs. And this is something which was done a lot in gene expression, in microarrays long time ago. And it is possible. The problem is that these methods assume that the majority of features do not change in abundance between conditions. If they do, then you remove the actual biological effect. And this was just one example. It was actually a titration experiment where we actually expected systematic increase in intensity. And of course, if we normalize it, then there is nothing else is left, right? So this is not a good way of doing it. So it's good because it's more stable than a single standard. It counts for all the data processing steps, but is not applicable, for example, for SRM, where we target proteins because we expect them to change. So this will not be a good normalization. And then another one, which is even more aggressive, is a quantile normalization, where we equalize not just the medians of the boxes, but every other aspect of the distribution. So those boxes become completely identical. And this, again, has been used in microarrays, also very aggressive normalization. However, turns out that it can be a very nice normalization if we have a large number of standards. So for example, if you have SRM experiments with labeled reference peptides for every endogenous peptide, you can actually apply this normalization to the standards. And this, because we do not expect to have any biology in them. And so this is actually the case. So this experiment actually had labeled standards. And so if we do this after normalization, now we see that this, in fact, was a biological trend that we had in this time course. So quantile normalization I would only use primarily when applied to standards if we have many of them. So that's the only idea. And then, so the output is the volcano plot, which I just described. Or if we have many comparisons, we also do heat map like that, where the rows are proteins and the columns will be pairwise comparison. For example, time 1 versus time 0, time 2 versus time 0, 3 versus 0, and so on. And so red means significantly up, blue means significantly down, and black is no change. So you can kind of also look at the trends of the proteins across time. Okay, so how do we actually get to this point? And this is really probably the core of what MSSTATS is doing in many ways in terms of working with many data processing tools and helping the reproducibility across the tools, essentially to mitigate to some extent these big changes across users that we have seen in the beginning. And let me illustrate this. So we have two data sets here. This is a DDA experiment from the MaxQuant LFQ paper, and this is a DIA experiment. They're both controlled mixtures, but doesn't really matter. They're both published. And we have this data set processed with Skyline, MaxQuant, and Progenesis, and this data set processed with Skyline and SpectraNote. And these gray shapes here are the histograms of all the log intensities of everything in this experiment. So everything, every single intensity across all proteins, all runs, all transitions and or fragments and so on, it's all reported together in this histogram of intensities. And so the first thing you see, for example, if we focus on DDA, is that those histograms, they're not in the same place. For example, this is shifted left compared to this one and so on, meaning that already numerically, different tools report different results. By itself, it's not necessarily a problem because we're not interested in peak intensity, we're interested in changes between conditions, right? And if some tool quantifies the peak entirely by its area and others truncate the tails and only report the center, or others report height at apex, of course, numerically this will be different. But if we can compare the values across conditions, so that's still meaningful. So that's not a problem, it's just interesting to notice. Now here we have the count of values which are either numerically 0 or very, very close to 0. And you see that Skyline has some, Progenesis has even more, Max Quant doesn't have anything. Clearly, Max Quant has some internal cutoff, which is being applied. So now if you compare the number of features, so you have 203,000 for Skyline, 224,000 for Max Quant, 56,000 for Progenesis, and also big discrepancy, 6,000 for Skyline and 2 million for SpectraNote. So really big differences in terms of the number of features. And in terms of zeros, Skyline reports 1,000, Max Quant reports none, and here we have 600. So clearly, when the peaks are very low intensity and are in the regions of interferences and close to the limit of detection, this is where the tools report things most differently. So what we decided to do to make sure that we interpret these outputs correctly, we decided to not take them literally and kind of read between the lines and try to understand the intention behind the zeros, NAs, small values, and so on. And what we decided to do is to come up with this vertical cutoff, which is here. And it's a dataset specific cutoff. And essentially, we will say that below this cutoff, we don't trust the values. Call them 0, NA, small value, whatever you want, we don't trust them. Because presumably they're too close to the limit of detection, limit of quantification to be reliable. And this cutoff is completely empirical, so what we do, we find 99th quantile. Here we find the median, calculate the distance, and flip it to the left. So it's just by assuming the symmetry of this distribution. And so now this is what happens. So this is a representation of a data set for one particular protein. We have columns are the runs and rows are the features. So the columns are really the biology that we're interested in. So here we have different conditions. Within each condition we have different subjects. We may have technical replicate runs for each subject. And this is where really the experimental design plays out. The rows are more the technology, because we just happen to quantify proteins with multiple features. And the values in the table, these are log intensities. And so now we see that some of them have missing values. So we came from here, right? So now we have the data set for one protein that looks like that. What we will do next is to introduce additional NA, but now we will call them informative NA. So they are missing values, but not randomly missing, but missing because they are coming from low intensities. And in statistics, it's called censoring. So it means that it's not missing arbitrary, but it's missing with information that the values are supposed to be low. And essentially everything below this vertical line, we call them censored values. And so now what we can do, turns out that, well, this can be written in a fairly complex way, statistical models, but the bottom line is that this type of models are identical, conclusions from this type of models are identical to conclusions to a two-step procedure where we first summarize all the intensities in the run across features, so focus on the yellow part, and then we focus as a second step on the pink part. And so this is what we do. So first, we essentially focus on the yellow part. We ignore which run came from which condition, but we pay attention to missing values especially censored values, so because they are below this cutoff. What we can do then is to look at this table as essentially a two-way analysis of variance table, and we can summarize features across the runs to simultaneously account for censored missing values, and also do it in a way that is robust to outliers. And it gives us one number per protein per run, and then we focus on modeling whatever experimental design was for this study.
So, could you explain again what the difference between the censored values is, or how do you get the non-censored MMs?
Right. Actually, I kind of went fast. I apologize. So, censored means informative, right? Informatively missing. And everything which appears for all the tools except Skyline, everything that has either NA or 0 or below this vertical cutoff, we assume that it's censored. So, for example, max quant progenesis and spectronaut can only have censored missing values. They will not have any other, because we don't have any additional information. The exception is Skyline, because Skyline actually makes a distinction when things are missing at random versus missing informatively. So, for example, if you have an SRM experiment, scheduled SRM experiment, and your window is kind of off, so you see the peak coming, but you cannot quantify it reliably, then it's not low, but you still don't have the value. So then it will be NA, and we leave it as NA because it's not censored. But the vast majority of cases, we make it censored because most other tools don't make this distinction, at least not that we know of. And then most likely it's censored value, so we just use that. Yeah. And you see that's why, for example, Skyline has 1,000 very small values, but only 33 NA, right? Because those events are relatively rare. Okay, so just to summarize, we start with the technology. We have the summary of all the features in the run in one value, and then we work on the pink part of the study to describe the biology. And the key contribution here is to summarize it in a way which handles censored missing values and handles outliers. And I'm just looking at the time. I'll probably skip because it's fairly technical, but feel free to come to me later, and I can give you the math for this if you would like. So it's different than just taking the sum of these values. There's really modeling involved behind that. And so what I would like to do in the remaining time is just to show the evaluation and kind of like the previous case, show how good it is, but also show the limitations of that. First, how it works, right? So this was this IPRG study, which I talked about in the beginning of the hour, where we have 4 conditions and spiked proteins. And so this is just one example. This is a protein which was fairly abundant, so we have many features. The gray lines here, they are the peptide ions of this protein across all replicates and conditions. The blue horizontal line is our cutoff, below which we don't trust the values. So MSSTATS assumes that everything below that is censored. We will not use them literally. So now linear model, essentially it's like a t-test. So we will take some kind of means of all of these features, including the outliers and so on, look for difference between the groups. So condition 1 versus 2, true-false change was 7.5. Linear model was clearly misled by these outliers, found 29-fold change, 20-fold change of 29. So clearly it was affected by these outliers. A very common summarization, and this is what Skyline is doing, is to take the sum of all the feature intensities in a column on the original scale and then take the log. This is a good way to do this, especially if you have zeros and some interferences are among small intensity values. It doesn't affect the summary very much, but it's very sensitive to interference in high intensity values because those will dominate the quantification. And it's kind of what you see as well. So here we have some interference in high intensity features, and it really resulted in the not being able to detect the difference between the 2 conditions. Tukey median polish, it's just a robust summarization, so it takes care of outliers but not of missing values, so it's getting closer. And what we do is we incorporate censoring and use also robust summarization, and it's the closest to the true fold change. So essentially, when all the features are good quality and don't have these issues, you can do anything you want and it will be fine for summarization. But our contribution comes from situations when we have a lot of cases like that. And so now coming back to why it matters, and remember I was talking from the beginning about reproducibility and like depending as little as possible on the choices that different tools make. And this is the summary of that. Here we have, for DDA, we have 3 data sets. They're all 3 controlled mixtures. We know for each of them what were the true changes. And now each data set was processed with multiple tools. And we look here, for example, using log of the sum quantification, the intersection between true changes which were detected by all the 3 tools. And the same thing compared to MSSTATS. And what we see is that consistently the intersection between the tools is higher with the approach by MSSTATS than with the approach by log sum. Meaning that if we don't take these values literally and we work with censoring and we take care of outliers, we can mitigate to some extent the discrepancies between different tools, and we have more consistency of the results regardless of which tool we used. And it's not like everybody has to use many tools, but to know that we will have more reproducibility and that our results depend less on the tool that we use, I think it's important to have. So this is a good thing, and the same thing for DIA. We have 2 controlled mixtures, and the same thing, we increase the consistency between the results. So this is very useful to have. And and I just forgot to say that then we use all of this data we just analyzed, we estimate the variance components, and we calculate sample size. That's the end of the MSSTATS part. Okay, so that's what happens here. Just maybe to wrap up with MSSTATS, it's used. We have a lot of downloads from the Skyline external tool website. It's distributed through Bioconductor. It's updated fairly frequently, so it may be useful to use development version sometimes. We have a message board where we try to be responsive as much as we can. So we do our best. And there's a website, so it's msstats.org, and we have different things here that you can look at, the documentation, case studies, and data sets, and so on. Okay. But so now I wanted also to kind of highlight the fact that what Brandon always likes to say is don't just do statistics, look at the data, right? So look at the individual peaks, don't just go with big summaries. And I think it's a really important point, because as hard as we try to make our conclusions independent of the tools, you know, garbage in, garbage out, right? So if we have features which are not very informative, it's very hard to make them informative somehow, right? And this is quite important. So essentially what I'm saying is that data processing matters. So even though we help, it doesn't mean that we can replace, you know, the steps that Skyline is doing or other tools are doing. And here's an example, so also an illustration of that. This is a data set with 10 time points in yeast, the one when I showed when we talked about normalization. So this was yeast samples across 10 time points where yeast was subjected to a particular stress, and the proteins were targeted to see how they respond to the stress across time points. So 6 time points, in this case 3 replicates per time point, so total 18 DIA runs on triple toe 5600. And then this data set was analyzed by Skyline in 2 ways. So the first, for the proteins, Skyline kept all peptide precursors and then kept top 6 fragments per peptide. But the second way was much more sophisticated. Before doing any of that, there was a pilot experiment on some of the yeast samples done just with technical replicates or biological replicates of the same condition to understand how much variability there was in different proteins. And so Skyline only quantified the targets which had low CV in this prior pilot experiment. So kind of using it more in a targeted manner, saying, let's first run the pilot study, see which targets are actually good quality, and then only quantify those in the actual study. And so now this was the procedure. So separate refinement experiment with 8 DIA runs, only include peptide precursors detected in more than 50% of refinement runs with CV less than 20%. After that, everything was the same, processed with some tools, and then we used Q-value filter cutoff and MSSTATS. And so this is the first comparison of everything versus low coefficient of variation. And you see already, of course, the number of proteins is smaller. We lost some 400 proteins by only quantifying features with good quality. And now we see also everything comparing to time point 0. Clearly, this identifies more differentially abundant proteins. But if you look at the biology of this, not many changes, very abrupt change, going up, going down, going up. That's less likely to be biologically plausible than something which has smaller change, more change, more change, more change. These were proteins which were all targeted because they were affected by the stress. And so this type of seesaw pattern seems to be due to an artifact more likely than a consistent increase in the number of differentially abundant proteins. So now I'm just focusing on this comparison, time point 60 versus 0. And we find that many differentially abundant proteins with quantifying everything, and we find that many going for low CV. And many of them were the same, but we found some new here and some other here. And this is what the volcano plots look like. So low CV has a kind of stronger pattern here a little bit, but not by much. So now what is interesting is to actually, okay, so that already, right, low CV versus everything already has substantial implications, right, on what we report. Now we can look at individual proteins. So this is a protein which was not found changing by any analysis, right? It's kind of found constant everywhere. So this is what happens when we have this protein with everything, and this is what happens when this protein is quantified with low CV features. So there are still some outliers, but I would argue that this is much easier to take care of by MSSTATS than this, because there are just too many, right? And this is clearly an outlier here. And so we see that there's differences because there are so many outliers. The p-value is borderline. Depending on your cutoff, you can actually claim that there is some change between 0 and 60. Here, it's much less evidence for that. And so now we find proteins which is changing only with low CV, but not with everything. And so again, comparing 60 versus 0. So here it kind of looks like a jagged, kind of a little bit up and down and more outliers. But here it's much more consistent and steady increase in time, which is also more biologically plausible. And you don't have these outliers, so you kind of see better the actual trend. And now we can also look more specifically what happens with different processing tools. So it was processed with Skyline, we keep low CV, and then also Spectronaut and DIU-Empire. The numbers are clearly very different. By the way, I don't mean that DIU-Empire doesn't do well. So this was an experiment of lower mass resolution, and DIU-Empire is not designed for that. It does much better on kind of more high resolution instruments. So it's not to say that it's not a good method. But clearly, the type of processing tool, even with MSSTATS, still affects the results. And so my message is that it's important to decide upfront what would be the protocol of the study and what we think is the most plausible approach here, and stick to this and not change the tools until we get the result that we like, essentially, right? And here I would argue that Skyline low CV is probably the best approach, even though it quantifies maybe a smaller number of proteins. But I think in terms of number of changes and their pattern across time, I would say it's the most biologically plausible. And it is so because it's using more information. It uses this extra pilot study, which tells us which fragments are the best to quantify. And so now if we kind of zoom into individual protein, it was the same protein as before. So this was the Skyline low CV, and we see that the actual values vary, the outliers vary, and therefore the conclusions will vary as well. So even though MSSTATS helps some, looking at the data and good quality data processing still matters quite a bit, so we cannot really replace that. Okay. Oh, and this is really kind of going back to this message saying that, you know, this type of things, the choice of the processing tools, the choice of normalization, the choice of the analysis strategy, it is something which is kind of hard to put in math, right? So it's our protocol of how we do the study, but it's kind of hard to incorporate the uncertainty of these choices in the p-value, right? And what I see often is that people say, okay, my p-value cutoff doesn't show my protein significance, so there's a problem. Well, there are so many steps which led to this p-value, and things could fail in so many ways that it's really important not to add uncertainty by changing the methods or changing strategies or changing parameters of the workflows and keep things very consistent. And it's like that in every area of science. And so here's a article in the Journal of Psychology, which essentially says undisclosed flexibility in data collection and analysis allows presenting anything as significant, right? So if we play with enough of those tools and cutoffs and parameters, then we can always show what we want. So we shouldn't be doing that. Okay, and so now I'm pretty much out of time, so let me just highlight what we do for this part of development of assays. And there are really two things I wanted to show. One is assay characterization, and Andy talked a lot this morning about these straight lines that we try to fit and that how they're actually not straight usually. And that's what we notice. And for large-scale studies where it's not possible to spike in standards always in the linear portion of the dynamic range, we essentially do, we have an approach to fit this, what we call a canonical calibration curve, characterize the uncertainty in this change point, and it gives us a nonlinear curve fit in a way that we can demonstrate that it quantifies the limits of detection and limits of quantification in a more accurate way than just by trying fit the straight line. I'm happy to tell you that during the break if you would like. And the second thing, as Andy mentioned as well, if we ask for replicates and if we need to collect replicates from many runs, it also means that it takes a long time. And there are many reasons for instruments to break down or our workflows to break down. And so what we also offer is a collection of tools for statistical process control, where you monitor standard mixtures or some kind of internal spiked standards across time to look for deviations from expected performance. For example, if you start having retention time drifts or mass shifts, you know, or other things. So there is a large body of literature, in statistical literature in this area. It's called statistical process control. And we implemented many of these tools here. And we even have a graphical user interface, so you can use it with that. And again, the input will be tabular or readable format, kind of like an export from MSSTATS. And we have summaries like go, warning, or stop when there are some violations of the specified ranges. Again, I can talk to you about this offline. So essentially, this is my very last part. Everything we talked about from the very beginning of the afternoon is all the ways which we need to consider to ensure reproducibility and the accuracy of the results. And so reproducibility, it's actually not a thing. It's a spectrum which goes from publication only to repeat the data analysis as they are, repeat the same data analysis and get qualitatively the same results with different tools, repeat the experiment on the same biological samples, or repeat the experiment on new biological material. And this is our gold standard. And we really want to be able to collect new samples on the same problem, run it with a similar workflow, analyze it with a similar way, but not necessarily identical way, and get qualitatively similar results. So that's really the gold standard for us. And so the software helps on this side. We can document and automate workflows and make them transparent. Data analysis helps on this part so that we can repeat the analysis with different tools and be less dependent on that. And of course, for this is the experimental design, selection of replicates, and standard operating procedures. So that's really the important part. Okay. Last thing, I want to thank many people. A lot of the MSSTATS work was done by my group, and especially Mina Choi, who is the lead on that part, and also Tsung Heng is a and Ting collaborators on that. Cyril did the work on assay calibration, and Eralp did the work on statistical process control. There was a whole team of IPRG who worked very hard on this study, including Brandon, who joined and did a lot of work on Skyline for that. A lot of what we do, we are really in debt to the Skyline team and McCoss Lab, because these are very useful interactions to us. We learned a lot, especially from Brandon and his development style. So he gives us a hard time, and we learn from that. And a lot of the collaborations and experiments came in a collaboration with the ETH.